Hello class, it's your fellow classmate Carolyne Fernandez, and today I'm going to be talking to you about the organism called Toxoplasma gondii that is very well known for the disease that it causes called toxoplasmosis. So let's begin.
Some general information about this organism is that it's a protozoan coccidian parasite. Its species is Toxoplasma gondii, and there's only one species of the genus, which is Toxoplasma. There are small mutations of it, but it's just one species. It's a one-cell eukaryote, and as you can see from the picture, it has a nucleus, just like how human cells do, a plasma membrane, and an ER. The disease which it causes is called toxoplasmosis, which affects the central nervous system. This organism wasn't discovered until 1908 by scientists named Nicole, Manciux, and Splendor, and this is while they were working on viruses. It wasn't really looked at and considered medically important, and it wasn't until 1970 that they finally discovered the full cycle of the organism.
What does it mean that this organism is an obligate intracellular parasite? This means that it's only capable of growing and reproducing inside the cells of its host, which are cats. A question that this research has made me think about, which I also want the class to think about, is how does Toxoplasma gondii recognize its final host, the cat? I will be providing the answer to this question at the end of my presentation.
Here we have a picture of the life cycle of the parasite and how it infects humans. We can see how felines here are the only definitive host and the only animals that can pass effective oocysts in their feces. Here we have the three infectious stages and morphological forms, which are sporocytes, tachyocytes, which causes acute infection, and bradiocytes, which causes chronic infection. So let's take it from the top. Our cat here gets infected with Toxoplasma gondii. The time within this feline is really important. Toxoplasma develops in the intestinal cells of the feline. It reproduces and then it's spread to the tissues, bloodstream. The organism is going to pass out in the form of feces from the cat. At first, it's going to be on sporulated cyst, and then it's going to go through sporulation that typically takes 3 to 4 days. And from there, they're going to be sporulated oocysts. And that's when it gets bad. It's transmitted through the fecal-oral route to others like livestock and humans. There are three routes of transmission, which is foodborne, zoonotic, and congenital. Foodborne is through the consumption of vegetables that are not properly washed, and then it's consumed by animals or humans. You can also get it through the consumption of undercooked meat from the intermediate host, which are the cows, the pigs, the sheep. Humans can easily get infected through their own pets, their own cats. That's why it's important for cat owners to clean their cat litters and not forget to wash their hands when they're making food because they can get infected. This parasite, being zoonotic, means that it can infect multiple different species at the same time, just like how you can see it in the picture. The other transmission route is congenital, which means infants can acquire the disease through transplacental infection. It will cross the placenta and infect the infant. And this is done by the consumption of undercooked meat, contaminated water, and that's why the doctors warn expecting mother on the seriousness of being extra careful if they own cats, and about consuming undercooked food. Another thing that I want to mention that I found pretty interesting is that tachyocytes here, they're going to multiply rapidly. It's sensitive to drugs and they will switch to bradiocyte if conditions are unfavorable. Bradiocytes are slower and develop cysts. A decrease in the body's immune response will make the bradiocyte go back to its tachyocyte form.
Most T. gondii infections are going to be asymptomatic in immunocompetent hosts. Those who are immunocompromised will get symptoms as the parasite moves from the blood to the tissues where it becomes an intracellular pathogen. In acute toxo, the initial presenting symptoms are nonspecific and more like the common cold. You'll get fevers, chills, fatigue. In chronic toxo, you'll get the acute symptoms and more. It involves mainly the central nervous system and the liver. Patients will get chorionitis that will cause blindness. Congenital transmission might cause the mother to have a miscarriage on the first trimester. Overall, it causes severe disease in the child. During the second and third trimester, the infant may have encephalitis and it might have other effects. There are different ways in which you can get diagnosed for toxoplasmosis. In serology, we're looking for the increasing numbers of antibodies. Titer going up is an indicative of an active infection. Tests are done to see if an infection is consistent with recent or prior exposure. If a patient has lesions in the central nervous system, that's when we would do a radiological evaluation like a CT or an MRI. A patient will get an ultrasonography and amniocentesis and PCR, which looks for specific changes in the fetus. The patient can also get a biopsy to see if they have tachyocytes. Immunocompetent patients are not going to have significant disease. It may resolve spontaneously. Immunocompromised patients will be given antiparasitics, a high dose of pyrimethamine sulfidazine, for 4 to 6 weeks. This is something not given to expecting mothers because it's a teratogenic for them. That's why they have to be extra careful during pregnancy. Immunocompromised patients will need treatment indefinitely. Those who are affected by the central nervous system are going to be given trimethoprim sulfamethoxazole. A human vaccine for toxoplasmosis does not exist. There isn't a multi-antigenic for the different growth stages in the life cycle that this organism has. Toxovax is available for veterinary use only. It prevents congenital toxoplasmosis in sheep. This vaccine is not suitable for human use, due to that it has adverse effects and may revert to a pathogenic strain. Toxoplasmosis is the leading cause of death attributed to foodborne illness in the U.S. The CDC states that 11% of the population in the U.S. 6 years and older have been infected with Toxoplasma. Worldwide, over 1 billion people are estimated to have been infected with it. From this picture, you can see that infection is highest in areas in the world that have hot, humid climates and low altitudes. This is because oocysts survive better in this environment. Based on my research, I predict that there won't be any future threats to this disease to human health. I think that it will continue to exist and cause damage in the immunocompromised population, but a vaccine will likely be developed. To wrap this up, I will answer the question asked earlier in the presentation. How does Toxoplasma gondii recognize its final host, a cat? From the research, I learned that it's because of the abundance of linoleic acid, which is a fatty acid that cats have. Most mammals have an enzyme called delta-6 denaturase that cats lack. Because of this, cats accumulate linoleic acid to higher levels than any other mammal. Toxoplasma gondii senses linoleic acid.
Thank you for listening to my presentation on Toxoplasma gondii.